Okay, good afternoon, and thank you for joining in on this virtual conference. My name is Ramani, and I have been a forensic scientist with the DNA Profiling Laboratory at Health Sciences Authority in Singapore for 8 years. Today, I will be talking on the topic, doing more with less. But first, a disclaimer. Before I start on today's topic for sharing, I would like to give a short introduction to who we are and what we do. We are the sole national accredited forensic DNA laboratory in Singapore. We have been accredited with ASPLAT Labs since 1996 and recently transitioned over to ANAB. As part of the human identification services we provide to law enforcement, our standard DNA profiling workflow involves semi-automated extraction using Promega IQ chemistry on the Maxwell FSC, quantitation using Quantifiler Duo on the QuantStudio 7, PCR using Global Filer on the ProFlex, and capillary electrophoresis on the 24-cap 3500 XL. We then issue our final reports, which are then tendered in court. Here are a selection of our publications on procedures we have developed or validated for use in casework. For investigative purposes, we used emergent technologies which have the potential, but we feel are not yet ready for use in court. We have both massively parallel sequencing platforms in place and use them for prediction of biogeographic ancestry, externally visible phenotypes, and whole genome mitochondrial DNA sequencing. We use the QIAGEN pyromark for pyrosequencing to predict age by looking at the proportion of methylation at 3 predictors, ELOF L2, TRIM59, and KLF14. We have also validated the use of the rapid hit system for urgent cases back in 2015. In forensic casework, the samples that we deal with are mostly compromised, with questionable quality and typically very limited quantities. The forensic community is continuously working to address these limitations. In line with this, we are currently evaluating the new Promega bone DNA kit. It has shown promising results for use with difficult body remain samples, such as those seen in DVI situations. However, this will not be the focus of today's sharing. The basis of doing more with less relates to being able to get more value, more data, more savings in terms of costs and our time with less input. Today, I will be sharing with you 2 methods which we have developed to be able to do more with less. I will start off with microFLOQ direct amplification first. The Copen microFLOQ direct swab is one which was specially designed for direct sampling and amplification. What is special about this micro swab is its small nylon fiber tip, which has been pre-treated to allow for in situ lysis. It also has a breakage point to allow the tip to be snapped off directly into a PCR tube or plate for direct PCR amplification. We use the microFLOQ swab by lightly moistening the tip with deionized water, applying the swab perpendicularly to the stain in question, giving the swab a few twists, and then returning the swab to its holder. Subsequently, the swab head is snapped off into a PCR tube containing the Global Filer master mix and processed as per our standard workflow. Given the small size of the nylon fiber tip, you can see how little sample is consumed, thus leaving sufficient sample for subsequent testing. The benefits that this method of sampling and direct amplification has offered are multifold. As mentioned, there is minimal sample consumption with sufficient sample left for subsequent testing. It is an easy-to-do procedure with almost no additional training, no additional equipment, and easily integrated in our existing workflow. Because we are skipping the extraction and quantitation steps, we are able to turn around results in 3 hours, which is especially useful for urgent cases. Furthermore, unlike rapid DNA systems, which use either 1, 3, 5, or 7 sample cartridges, and any additional samples would mean additional 90 minutes or under 2-hour runs. Again, depending on which rapid DNA system you're talking about, the use of microFLOQ direct amplification allows us to process up to 20 samples and controls in the 24-cap CE system to be run in the same 3 hours. This is also cheaper than standard processing by 14% and up to 20 times cheaper per sample compared to rapid DNA systems. Some laboratories would have concerns with direct amplification due to requirements for consensus-based reporting of mixed DNA profiles, or with capillary electrophoresis artifacts due to over-amplification. We too had these issues and sought to see how we could improve on this method. To address these concerns, we realized we needed to generate a lysate. So instead of snapping off the microFLOQ head into a tube containing our master mix, it went into some low TE buffer and a short incubation to allow the lyse cell contents to enter the buffer. This provided us with sufficient volume for multiple amplifications using global filer and the option of varying the amount of lysate used for amplification. We evaluated this modification with blood stained on a wide variety of substrates and reported on the performance of this compared to our standard sample processing of cotton swabs of the blood stains on the different exhibit types. These box plots show the range in median peak heights in the DNA profiles obtained from blood on the different substrates. Straight away, we can see that there was much wider variation in RFU observed with modified microFLOQ procedure because of the natural variation in template use for amplification because no quantitation was performed compared to the standard method. Overall, we obtained complete DNA profiles for the modified microFLOQ across all the substrates with well-balanced sister peaks and the majority of peaks above our stochastic threshold. In the last two substrates, we observed that the modified microFLOQ workflow was able to generate complete DNA profiles where our standard workflow failed to produce any reportable peaks. Here, we see an excerpt from the respective electropherograms. In the standard processing, there was no inhibition detected during quantitation. The internal positive control CT values were no different from other non-inhibited samples. We believe that the humic acid in the soil may have competitively sequestered the released DNA during extraction, and hence, there was no DNA eluted. On the left are photos showing what blood on the soil-stained fabric and rock looked like. And so, in addition to the benefits highlighted earlier, and with a slight increase in processing time, we have also addressed the concerns of CE artifacts due to over-amplification and gained the ability to do replicate amplifications to allow consensus reporting of DNA profiles. By adopting this modified microFLOQ direct amplification method, we have been able to do more with less. Moving on, I will next be sharing about ancestry prediction using Y-STR DNA profiles. We have been doing prediction for biogeographic ancestry using ancestry informative markers genotyped on the Illumina massively parallel sequencing platform, using custom reference populations for the predominant Chinese, Malay, and Indian populations in Singapore, and using the online snipper tool. This has allowed us to predict ancestry with an accuracy of about 90%, and the ability to detect admixture between these ancestry groups when present at a 1:1 up to a 3:1 ratio. However, this capability required additional sample consumption, significant costs, and a 4 to 5 day wait for the MPS results. We know that Y haplogroups are biogeographically linked, as you can see by the multiple colors associated with the different regions of the world. In our laboratories, we have already been performing Y-STR DNA profiling for a number of years, and for increasingly more case types. Analysis of Y-STR profiles generated in our laboratory over the past few years showed that we had about 66,000 unique haplotypes from about 78,000 Y-Filer plus DNA profiles. The frequency distribution of these haplotypes show that the majority, nearly 3/4, are seen only once, otherwise known as singletons. We use Shannon's entropy as a measure of information content between our populations in the tested Y-STR loci. This is influenced by the proportion of alleles at each locus if you look at the formula. We observe that the Y-STR loci are quite rich in information content. From the bar plots, DYS385 and DYF387S1, both of which are multi-copy markers and so have the most number of alleles among the tested loci, also have the highest information content. We looked at linkage disequilibrium across the Y-STR loci and observed that despite inclusion of rapidly mutating Y-STR markers, the entire set of Y-STR loci were all very strongly linked. The high degree of shared information that we saw in the previous slide, combined with the strong non-recombining properties of the loci and the generally reported low occurrence of mutation at between 10 to the -4 and 10 to the -2 per generation, meant that there was preservation of ethnicity information in the Y-STR profiles, thus allowing them to be used for ancestry inference. We decided to use machine learning algorithms to model the differences in our local Chinese, Malay, and Indian populations. We didn't know how the Y-STR would behave and didn't know which ones would work the best. And so we tried to cover most classes of machine learning algorithms, such as support vector classification, tree-based modeling with different boosting techniques, neural networks, and k-nearest neighbors. We further supplemented these algorithms with 2 additional classifying algorithms, which took input from the latter 4 models and gave a prediction either based on the majority, if you look at the voting classifier, or based on neural network modeling, if you look at the stacking classifier. While each of these machine learning algorithms had slightly different accuracies, they were generally able to lead to the same predictions. 5 different algorithms leading to the same answer reinforced that the predictions were accurate at around 90% accuracy. And this was reiterated by the 2 classifier algorithms. So effectively, because we had already been performing Y-STR DNA profiling on increasingly more case types, we were also able to do ancestry prediction using machine learning without any additional sample consumption, additional costs, or the typical 4 to 5 day wait for MPS results. And so we have succeeded in doing more with less. To continuously improve on our capabilities following this theme, we hope to expand the use of the modified microFLOQ technique to include semen stains, and as well as to expand the use of the machine learning algorithms for ancestry prediction in other Asian populations. With that, I come to the end of my presentation. I would like to acknowledge HSA, the organization which I work for giving us the backing and opportunity to continue to be the National Forensic DNA Laboratory. Thermo Fisher for giving us the chance to share with all of you today. and everyone from our laboratories who have contributed to the research and developments mentioned so far.